Chronic stress leads to neuronal fatigue and, eventually, a disruption in blood flow to the white matter.
The brain is already losing function before you notice anything is wrong. Electrical signals that normally move in coordinated patterns begin to fragment under sleep loss. Regions that should activate together start firing out of sequence, while others briefly go silent and then return. This is not simple fatigue. It is a loss of synchronization across neural networks, where communication between brain regions becomes unstable even while you remain awake and functional. These disruptions occur without warning because the brain does not signal when its internal coordination begins to fail. You can still think, move, and respond, but continuity is already breaking down beneath awareness. The prefrontal cortex, responsible for decision-making and attention, shows inconsistent activation. At the same time, deeper structures like the thalamus lose timing precision, creating a mismatch between regions that are supposed to operate in sync. The result is fragmentation, not total failure, which makes it invisible to perception. This instability is measurable. Functional imaging shows reduced connectivity between brain regions during sleep deprivation. Networks lose coherence, meaning signals no longer travel efficiently. But this is not only an electrical problem. Beneath the disrupted signaling, blood flow regulation begins to fail. The brain depends on precise delivery of oxygen and glucose, but under sleep loss, this system becomes inconsistent, creating areas that receive less supply than they require. When oxygen delivery becomes uneven, certain regions begin operating under stress. Neurons depend on continuous energy to maintain function, and even small deficits can disrupt their stability. Some areas receive delayed blood flow, while others receive less than needed. This imbalance contributes to the network instability already present, pushing the brain closer to structural vulnerability rather than just temporary dysfunction. Small blood vessels deep within the brain are especially sensitive to these changes. These microvessels supply white matter and subcortical regions and have limited backup pathways. When regulation becomes unstable, these vessels are exposed to fluctuating pressure and inconsistent signaling, increasing the likelihood of brief interruptions in blood flow. These are not large blockages, but small, transient disruptions that occur at a level too subtle to immediately detect. This is where the condition becomes defined. These events are called silent strokes. They are small areas of brain tissue that lose blood supply long enough to sustain damage, but not long enough to produce noticeable symptoms. The term silent does not mean harmless. It means the damage occurs without awareness, allowing it to accumulate without interruption. Each of these micro-events affects a small cluster of neurons. Oxygen delivery stops, energy production fails, and the cells begin to lose function. Because the affected area is small, the brain compensates. Other regions adjust, signals reroute, and overall function appears preserved. But the damaged neurons do not recover. Once they cross a threshold of failure, the loss becomes permanent. This process can begin during sustained sleep loss without requiring extreme conditions. The longer the brain remains in this unstable state, the higher the probability of repeated microvascular interruptions. There is no immediate feedback mechanism to stop it. No pain, no clear symptom, no signal that structural damage is already forming beneath normal awareness. This creates a critical disconnect. What you feel is fatigue, slower thinking, or reduced focus. What is actually happening is micro-level injury inside the brain. The instability you experience is not the damage itself, but the environment that allows the damage to occur. The actual injury happens silently, inside small vessels and localized clusters of neurons that fail without detection. This is how the process begins, not with a sudden collapse, but with gradual fragmentation of network function, followed by instability in blood flow, then brief interruptions, and finally loss at the cellular level. Once neurons are damaged beyond recovery, they are not restored. The brain adapts around the loss, but the original structure does not return. The instability does not remain confined to signaling. As sleep loss continues, the regulation of blood flow becomes progressively unreliable. The brain depends on continuous adjustment of vessel diameter to match metabolic demand, but this coordination begins to break down. Instead of precise responses, vessels constrict or dilate inconsistently, creating fluctuations in perfusion across different regions. This loss of control disrupts neurovascular coupling. Under normal conditions, active neurons receive increased blood supply within milliseconds. During sleep deprivation, that response is delayed or incomplete. Some regions activate without receiving sufficient oxygen, while others receive blood flow without corresponding activity. The timing mismatch becomes a structural risk, not just a functional inefficiency. These fluctuations create pockets of vulnerability. Areas with high metabolic demand, such as the prefrontal cortex and hippocampus, are especially sensitive to even slight reductions in oxygen delivery. When supply becomes inconsistent, neurons in these regions begin operating under suboptimal conditions. Energy production becomes unstable, and cellular processes start to degrade. The instability is not constant. It shifts across the brain in an uneven pattern. One region may experience reduced flow for a brief period and recover, while another region becomes affected. This moving pattern of perfusion imbalance prevents the brain from stabilizing its internal environment. It remains in a state of continuous adjustment without reaching equilibrium. Deep white matter is particularly exposed to this process. These regions rely on long, narrow vessels with limited redundancy. When blood flow drops, there are few alternative pathways to compensate. Even short interruptions can create localized stress, especially when repeated over time. These areas are not highly active in conscious processing, which makes early damage difficult to detect. At the level of small vessels, structural stress begins to build. Endothelial cells lining the vessels are affected by fluctuating pressure and irregular signaling. Their ability to regulate vessel tone weakens. This increases the likelihood of transient narrowing or brief occlusion, especially in already vulnerable regions. These micro-level disruptions do not produce immediate infarction every time. Many are temporary and resolve before causing permanent damage. But the frequency of these events increases under sustained sleep deprivation. The brain is repeatedly exposed to short periods of reduced blood flow, creating cumulative stress on both vessels and neurons. Carbon dioxide regulation also becomes less stable. Normally, CO levels help control vessel dilation. During sleep loss, this regulatory system becomes less precise, further contributing to unpredictable vessel behavior. Blood vessels may constrict when dilation is needed or fail to respond when oxygen demand increases. This creates a condition where perfusion is not only reduced, but also poorly timed. The brain does not simply receive less blood, it receives it at the wrong moment. This timing failure amplifies the risk of neuronal stress because energy supply no longer matches activity patterns. Inflammatory signaling begins to increase as well. Sleep deprivation is associated with elevated inflammatory markers that affect vascular function. These signals can impair endothelial responsiveness and promote subtle changes in blood flow dynamics. Over time, this contributes to a less stable vascular environment. The combined effect is a system that cannot reliably maintain oxygen delivery. Some neurons receive enough supply to continue functioning, while others operate at the edge of failure. This uneven distribution creates zones where damage is more likely to occur if flow drops even slightly further. These zones are not fixed. They shift depending on activity, metabolic demand, and the current state of vascular regulation. This makes the process unpredictable at the level of conscious experience. There is no single area that consistently fails, which is why symptoms do not appear early. Instead, the brain maintains overall function by redistributing workload. Regions that remain stable compensate for those that are temporarily impaired. This masking effect delays detection, but increases long-term risk because underlying instability persists. As these fluctuations continue, the probability of a critical interruption increases. A vessel that is already constricted or poorly regulated may briefly close or fail to deliver sufficient blood flow. If the interruption lasts long enough, even on a small scale, it can initiate tissue damage. This is the transition point from instability to injury. The interruption does not need to be prolonged. Even short durations of insufficient blood flow can disrupt cellular processes. Neurons require continuous oxygen and glucose to sustain membrane potentials and synaptic activity. When supply drops below a critical threshold, function begins to fail rapidly. At first, the effect is reversible. If blood flow returns quickly, neurons can recover. But repeated exposure to these interruptions weakens their resilience. Each event reduces the margin of safety, making future interruptions more likely to result in permanent damage. This is how silent strokes begin to emerge from vascular instability. They are not isolated events. They are the cumulative result of repeated perfusion failures across small regions of the brain. Each event may be minor, but together they create a pattern of structural injury that builds over time. The absence of immediate symptoms allows this process to continue unchecked. The brain does not register these small areas of damage as urgent signals. Instead, it adapts by reallocating function, maintaining outward performance while internal structure changes. This adaptation has limits. As more regions experience intermittent stress, the capacity for compensation decreases. Networks that were once stable become dependent on fewer functional pathways. This increases vulnerability because there are fewer alternatives when disruption occurs. At this stage, the brain is no longer operating with full redundancy. It is functioning with reduced resilience, even though conscious awareness remains intact. The instability in blood flow is no longer just a background condition. It has become an active driver of structural change. Each fluctuation carries risk. Each interruption has the potential to convert temporary dysfunction into permanent loss. And because these events occur at a scale below perception, they accumulate without interruption, setting the stage for the next phase of damage. Silent strokes are not defined by how they feel. They are defined by what happens to the tissue. A silent stroke is a small area of brain tissue that loses blood supply long enough to sustain injury, but not long enough to produce immediate, recognizable symptoms. There is no sudden collapse, no visible deficit, no clear event marking when it occurred. The absence of symptoms is not because the damage is insignificant. It is because of scale and location. These events affect small clusters of neurons, often in regions that do not directly control movement or speech. The brain can redistribute function around these areas, masking the loss while the structural damage remains. On imaging, these injuries appear as small lesions, often described as lacunar infarcts or microinfarcts. They are most commonly found in deep brain structures and white matter pathways, where blood vessels are narrow and more vulnerable to interruption. These areas are critical for connectivity, even if they do not produce immediate, obvious deficits when damaged. Sleep loss increases the conditions under which these events occur. As blood flow regulation becomes unstable, microvessels are more likely to experience brief occlusions. These occlusions may last only seconds or minutes, but that is enough to disrupt the cells they supply. When oxygen delivery stops, even briefly, neurons begin to lose their ability to maintain electrical balance. The key factor is duration. Neurons can tolerate short interruptions, but only within a narrow window. If blood flow is not restored quickly enough, cellular processes begin to fail. Energy production drops, ion gradients collapse, and the cell can no longer sustain its function. At this point, the damage becomes structural. Not every interruption leads to permanent injury. Some events resolve before reaching that threshold. But under repeated stress, the likelihood of crossing that threshold increases. Sleep deprivation does not create a single catastrophic event. It creates repeated opportunities for small failures to become permanent. These events are often clustered over time rather than isolated. One microinfarct may occur, followed by another in a different region hours or days later. Each event is small, but together they form a pattern of accumulating damage that slowly alters the architecture of the brain. Because each individual event is silent, the accumulation is also silent. There is no clear moment when the process begins. There is no single point where the brain signals that damage has reached a critical level. Instead, there is gradual change beneath stable outward function. This mismatch between structural change and perceived function is what makes silent strokes difficult to detect without imaging. Magnetic resonance imaging can reveal these lesions as small areas of altered signal intensity. They are often discovered incidentally, long after they have occurred. By the time they are visible, the tissue has already been affected, and recovery is limited or absent. The distribution of these lesions is not random. They tend to appear in regions supplied by small penetrating arteries. These vessels branch off larger arteries and travel deep into the brain without significant collateral support. When they fail, the area they supply has no alternative source of blood. This makes them particularly vulnerable during periods of unstable perfusion, such as prolonged sleep loss. The damage in these regions affects connectivity more than isolated function. White matter pathways carry signals between different parts of the brain. When these pathways are disrupted, communication becomes less efficient. The effect is subtle at first, often presenting as reduced processing speed or slight changes in attention. But these changes are not immediately recognized as structural injury. They are often attributed to fatigue or temporary cognitive decline. This misinterpretation allows the underlying process to continue without intervention. Silent strokes also differ from larger strokes in their progression. A large stroke produces immediate and obvious symptoms because it affects a significant portion of brain tissue. A silent stroke, by contrast, affects a small area but contributes to a larger pattern over time. The cumulative effect can eventually reach a threshold where symptoms become noticeable. But by that point, the damage is no longer isolated. It is distributed across multiple regions, making recovery more complex and less complete. Sleep deprivation accelerates this accumulation by maintaining the brain in a state of instability. Each period of insufficient rest prolongs the conditions that allow microvascular interruptions to occur. The brain does not return to a fully stable baseline, increasing the probability of additional events. This is not a linear process. It does not progress at a constant rate. Instead, it depends on the interaction between vascular stability, metabolic demand, and the duration of sleep loss. Some periods may produce no detectable damage, while others may result in multiple micro-events. The unpredictability adds to the risk because there is no reliable external indicator of when damage is occurring. What remains consistent is the lack of immediate feedback. Silent strokes do not produce pain. They do not trigger protective responses that force behavioral change. The brain continues to operate, adapting around the damage as it accumulates. This adaptation has a cost. Each new lesion reduces the efficiency of the network. Signals must travel through alternative pathways, increasing processing time and reducing precision. Over time, this leads to measurable changes in cognitive performance, even if the individual events were not noticed when they occurred. At the cellular level, the affected neurons do not recover once they have been damaged beyond a certain point. They may remain structurally present for some time, but their function is impaired or lost. Surrounding cells may attempt to compensate, but the original connectivity is altered. This is why silent strokes are not reversible events. They represent permanent changes in brain structure, even if their effects are initially subtle. In the context of sleep loss, they are not rare anomalies. They are a predictable outcome of sustained vascular instability and repeated micro-level interruptions in blood flow. Each event is small. Each event is silent. But each event contributes to a larger pattern of change that the brain cannot fully undo. Damage from silent strokes does not spread evenly across the brain. It follows patterns defined by blood supply and energy demand, which is why certain regions are affected earlier than others. The hippocampus is one of the first exposed because it requires constant oxygen to maintain memory formation. During sleep deprivation, blood flow to this region becomes inconsistent, and even brief reductions can disrupt its stability. What is experienced as simple forgetfulness or difficulty retaining information is often the surface effect of neurons struggling to maintain function under reduced supply. At the same time, the prefrontal cortex begins to show similar vulnerability. This region controls attention, decision-making, and behavioral regulation, and it operates under high metabolic demand. When perfusion becomes unstable, its activity fluctuates rather than failing completely. This leads to slower processing, reduced precision in judgment, and inconsistent focus. These changes are subtle and easily attributed to fatigue, but they reflect underlying stress in regions that are no longer receiving stable support. Deep white matter becomes affected through a different mechanism. Instead of controlling specific functions, it connects distant parts of the brain, allowing information to move efficiently between regions. The vessels supplying white matter are small and have limited backup pathways, making them sensitive to drops in blood flow. When microvascular interruptions occur, communication slows rather than stops. Signals still travel, but with reduced speed and coordination, creating inefficiency across the entire network. Subcortical structures are also involved early in the process. These areas regulate movement, emotion, and basic cognitive integration, and they depend on small penetrating arteries that are highly vulnerable to instability. When these vessels are affected, the result is not a single clear deficit, but a diffuse impact across multiple systems. Small changes in regulation accumulate, but they do not immediately trigger recognition as structural damage. These regions do not fail in isolation. Each contributes a small change that the brain initially compensates for by redistributing workload to other areas. Functional imaging shows increased activity in unaffected regions as they attempt to maintain overall performance. This compensation keeps behavior stable, but it increases metabolic demand in those regions, making them more vulnerable over time. As a result, the pattern of stress begins to spread. This spreading effect creates a cycle. Regions under stress become less efficient, other regions compensate, and the compensating regions gradually become unstable as well. What begins as localized vulnerability in memory and executive areas expands into broader network involvement. The brain remains functional, but it requires more effort to maintain the same level of performance, and its margin for stability continues to shrink. Certain anatomical zones are especially exposed during this process. Watershed areas, located between major arterial territories, naturally receive less consistent blood flow. Under normal conditions, they function adequately, but during sleep deprivation, they become high-risk zones for perfusion drops. Microinfarcts in these areas disrupt connectivity between larger brain regions, further reducing efficiency without producing immediate symptoms. The overall pattern is one of distributed micro-injury rather than isolated damage. The hippocampus affects memory stability. The prefrontal cortex affects control and attention. White matter affects communication speed, and subcortical regions affect integration. Individually, these changes are small, but together they begin to alter how the brain operates as a system. At this stage, the brain is still able to maintain outward function, but it is doing so with reduced redundancy. Fewer pathways remain fully stable, and more processes depend on precise conditions to function correctly. Continued sleep loss or additional stress increases the likelihood that these fragile systems will experience further disruption. This is how early regional damage transitions into network-level vulnerability. The brain does not collapse suddenly. It becomes progressively less efficient, less stable, and less able to compensate for new disruptions, even though no single event marks the change. The process now moves from unstable blood flow to direct cellular failure. When a microvascular interruption lasts long enough, neurons are no longer able to sustain the energy required for normal function. This is not a gradual decline at the cellular level. It is a rapid shift from stability to failure once oxygen delivery drops below a critical threshold. Neurons depend on continuous production of ATP to maintain electrical balance. ATP powers the ion pumps that keep sodium and potassium in the correct distribution across the cell membrane. An oxygen supply is reduced, ATP production decreases. As ATP levels fall, these pumps begin to fail, and the cell can no longer regulate its internal environment. The first consequence is loss of membrane stability. Sodium begins to accumulate inside the neuron, while potassium leaks out. This disrupts the electrical gradient that allows the neuron to generate signals. The cell becomes depolarized in an uncontrolled way, unable to reset to its normal resting state. Calcium begins to enter the cell in excess. Under normal conditions, calcium levels are tightly regulated because it acts as a signaling molecule. During ischemia, calcium influx becomes uncontrolled. This activates enzymes that begin to break down cellular structures, including proteins, membranes, and components of the cytoskeleton. At the same time, neurotransmitter release becomes dysregulated. Glutamate, the primary excitatory neurotransmitter in the brain, is released in excessive amounts. Instead of controlled signaling, there is continuous stimulation of neighboring neurons. This process is known as excitotoxicity. Excitotoxicity amplifies the damage. Overstimulated neurons consume more energy while already deprived of oxygen, accelerating their failure. Calcium influx increases further, and the destructive enzymatic processes intensify. What began as a localized energy deficit expands into a cascade that affects surrounding cells. Mitochondria, the structures responsible for energy production, begin to fail under these conditions. Without sufficient oxygen, they cannot sustain ATP generation. In addition, they produce reactive oxygen species as byproducts of impaired metabolism. These molecules damage cellular components, including DNA, proteins, and lipid membranes. Oxidative stress adds another layer to the injury. The cell is now facing energy depletion, ionic imbalance, excitotoxic signaling, and molecular damage simultaneously. These processes do not occur in isolation. They reinforce each other, accelerating the transition from dysfunction to irreversible injury. If blood flow is restored quickly, some of these processes can be halted. The cell may recover partial function, but there is a threshold beyond which recovery is no longer possible. When structural damage reaches a certain level, the neuron cannot return to its previous state, even if oxygen supply is reestablished. At that point, cell death pathways are activated. Some neurons undergo necrosis, where the cell loses membrane integrity and breaks apart. Others enter programmed cell death or apoptosis, where internal signaling leads to controlled dismantling of the cell. Both outcomes result in permanent loss of function. The surrounding environment is also affected. Neighboring cells experience increased stress due to inflammatory signaling and disrupted extracellular balance. This creates a localized zone of injury that extends beyond the initial site of blood flow interruption, although still small enough to remain clinically silent. Glial cells, which support neurons, respond to this damage by attempting to stabilize the environment. They clear debris and release signaling molecules that influence repair processes. However, their capacity to restore function is limited. They can contain damage, but they cannot replace lost neurons. This entire cascade can unfold within minutes of insufficient blood flow. It does not require prolonged deprivation to initiate. Short interruptions, if repeated, can cumulatively weaken cells and make them more susceptible to future injury. Sleep deprivation increases the likelihood of these cascades by maintaining unstable perfusion over time. Each microvascular interruption creates a new opportunity for cellular failure. Even if some cells recover from early events, repeated exposure reduces their resilience. The result is not a single wave of damage, but multiple small cascades occurring in different regions at different times. Each cascade contributes to a growing pattern of structural loss that the brain cannot reverse. Importantly, the brain does not regenerate these neurons once they are lost. Some limited plasticity allows for adaptation, but the original cells and their connections are not restored. The network must reorganize around gaps that remain permanently. This is the point where silent strokes become defined not just by vascular events, but by cellular death. The interruption in blood flow initiates the process, but the outcome is determined by what happens inside the cells. Once the cascade crosses the threshold of irreversibility, the damage is fixed. It does not depend on future conditions. It becomes part of the brain's structure. And because these events occur without immediate symptoms, they accumulate without interruption, each one adding another layer of permanent change. Damage does not occur as a single event. It accumulates through repeated microvascular interruptions that occur over time without detection. Each event may be small, but the brain does not fully reset between them. Residual stress remains in affected regions, lowering the threshold for the next disruption. After an initial micro-event, some neurons partially recover if blood flow is restored quickly. Electrical activity may return, and function appears normal. But recovery is not complete. Cellular reserves are reduced, and structural integrity is weakened. The neuron remains functional, but less stable than before. When another interruption occurs, the same neuron is now more vulnerable. The margin between survival and failure is smaller. What could have been tolerated previously may now cross the threshold into irreversible damage. This is how repeated exposure converts temporary dysfunction into permanent loss. This process creates layering. Each micro-event leaves behind a small area of altered tissue, even if function is preserved in the short term. Over time, these areas accumulate, forming a pattern of distributed injury across the brain. The damage is not concentrated in one location, but spread across multiple regions. Because each event is silent, there is no interruption in behavior to allow recovery. The brain continues to operate under stress, increasing the likelihood of additional events. There is no protective feedback loop that forces a pause or correction. The brain attempts to compensate for these accumulating changes. When one pathway becomes less efficient, alternative pathways are used. This redistribution maintains overall function, but it increases the load on remaining intact circuits. These circuits must process more information, often under the same unstable conditions. Compensation has limits. As more regions experience micro-injury, the number of available alternative pathways decreases. The brain becomes more dependent on fewer routes to maintain function. This reduction in redundancy increases vulnerability to further disruption. At the same time, communication between regions becomes less efficient. White matter pathways that have been affected by repeated micro-events transmit signals more slowly. Timing between regions becomes less precise. Coordination weakens, even if individual regions remain partially functional. This creates a state where the brain appears normal at rest but struggles under demand. Tasks that require sustained attention, rapid processing, or complex coordination become more difficult. These changes are often subtle and attributed to fatigue or stress, not structural damage. The accumulation is not linear. Some periods may produce multiple micro-events, while others produce none. The variability depends on the degree of vascular instability and metabolic demand. This unpredictability makes it difficult to correlate subjective experience with underlying structural change. What remains consistent is that each event contributes to a cumulative burden. The brain does not remove these lesions. It adapts around them, but they remain as permanent alterations in structure. Over time, the number of affected areas increases. As this burden grows, network efficiency declines. Signals take longer to travel. Integration between regions becomes less reliable. The brain must expend more energy to achieve the same level of performance, further increasing metabolic demand. This increased demand feeds back into the same cycle of vulnerability. Regions under higher load require more oxygen and stable perfusion, but the vascular system remains inconsistent under sleep deprivation. This mismatch increases the likelihood of additional microvascular interruptions. The system becomes self-reinforcing. Instability leads to damage. Damage increases demand. Increased demand amplifies instability. Despite this progression, outward function can remain relatively stable for a prolonged period. The brain is capable of masking a significant amount of structural change through compensation. This delays recognition, but does not prevent progression. There is no clear boundary where accumulation suddenly becomes visible. Instead, there is gradual erosion of efficiency and resilience. The brain continues to function, but with less capacity to handle additional stress. Eventually, the cumulative burden reaches a point where compensation is no longer sufficient. But before that point is reached, a substantial amount of structural damage may already be present. This phase is defined by accumulation without detection. The brain is changing at a microscopic level, event by event, without producing signals that would indicate the need for intervention. Each silent stroke adds to the total. Each recovered region carries residual weakness. Each compensated pathway increases load elsewhere. And all of it occurs without interruption, building a pattern of damage that only becomes visible after it has already been established. There is a point where compensation no longer holds. The brain can adapt around small areas of damage for a limited time, but as the number of affected regions increases, the system reaches a threshold where remaining pathways cannot sustain the load. This is not a sudden event. It is a transition where stability gives way to persistent inefficiency. Before this threshold, damaged regions are bypassed. Signals reroute and function is preserved. After this threshold, rerouting becomes incomplete. Some signals arrive late. Others degrade before reaching their target. Coordination between regions begins to fail in a way that can no longer be fully masked. This shift marks the beginning of measurable decline. Processing speed slows beyond what can be attributed to fatigue. Attention becomes inconsistent, not just reduced. Memory retrieval becomes unreliable, not just delayed. These changes reflect structural limitations rather than temporary functional variation. At the network level, synchronization weakens further. Regions that once operated in coordinated patterns begin to drift out of alignment. Timing differences increase, and the precision of communication decreases. The brain remains active, but its internal organization becomes less stable. White matter integrity plays a critical role at this stage. As microinfarcts accumulate in these pathways, signal transmission loses efficiency. Delays increase, and the coordination required for complex tasks becomes harder to maintain. This is not due to a single lesion, but the combined effect of many small disruptions. The hippocampus also begins to show sustained impact. Memory formation becomes less reliable because repeated stress has reduced the stability of its networks. Information may still be processed, but consolidation becomes inconsistent. This is not a temporary lapse. It reflects cumulative structural change. The prefrontal cortex loses its ability to maintain continuous control over attention and decision-making. Fluctuations that were once brief become more persistent. The region can still function, but it cannot sustain stable output over time. This leads to variability in performance rather than a uniform decline. At this point, the brain is operating with reduced redundancy. There are fewer intact pathways available to compensate for disruption. Any additional stress-continued sleep loss, metabolic demand, or further vascular instability has a greater impact because there are fewer alternatives. This is the irreversibility threshold. It is not defined by a single event, but by the cumulative loss of capacity to recover. Individual neurons that have been damaged do not regenerate. Networks that have been disrupted cannot be restored to their original configuration. A system can adapt, but it cannot return to its previous state. Once this threshold is crossed, further damage produces more noticeable effects. The brain can no longer fully compensate, so each new micro-event contributes directly to functional decline. What was once silent begins to become detectable. Importantly, crossing this threshold does not stop the process. Vascular instability may still be present, and microvascular interruptions can continue to occur. The difference is that their impact is no longer masked. Each event adds to an already compromised system. This stage represents a shift from hidden accumulation to visible consequence. The underlying mechanism has been active for some time, but only now does its effect become apparent at the level of behavior and cognition. Despite this, the damage remains irreversible. Restoring sleep or improving conditions may stabilize the system and reduce further risk, but it does not reverse the structural changes that have already occurred. Lost neurons are not replaced, and disrupted pathways do not return to their original form. The brain continues to function, but with reduced efficiency and resilience. Tasks that were once automatic require more effort. Stability depends on maintaining optimal conditions, because the margin for error has been reduced. This is the point where silent strokes transition from an invisible process to a measurable one. The accumulation has reached a level where compensation is no longer sufficient to maintain full function. And once this transition occurs, the system does not return to its original baseline. By the final stage, the brain is no longer operating with its original structure. The accumulation of silent strokes has altered the architecture of multiple regions and the connections between them. What remains is a system that functions, but differently, less efficiently, less reliably, and with reduced capacity to adapt. The changes are distributed. White matter pathways that once allowed fast, coordinated communication are now interrupted by multiple points of damage. Signals must travel through longer or less efficient routes, increasing delay and reducing precision. This affects not one function, but the integration of all functions. Cortical regions continue to operate, but their output is less stable because the inputs they receive are no longer synchronized. The prefrontal cortex processes information with reduced consistency. The hippocampus forms memories with less reliability. Subcortical systems regulate activity with diminished precision. Each region is still active, but the coordination between them has been permanently altered. This is structural finality. The brain has reorganized around loss, but it has not restored what was lost. The original network, with its redundancy and efficiency, no longer exists in its previous form. What replaces it is a modified system built on remaining pathways, constrained by the damage that has accumulated. At this stage, the risk profile changes. The presence of multiple microinfarcts increases the likelihood of larger vascular events. The same instability that produced silent strokes can extend to more significant interruptions in blood flow. The system is more vulnerable because its protective capacity has been reduced. The effects are not confined to cognition. Motor coordination, emotional regulation, and processing speed can all be influenced as more regions become involved. The impact depends on the distribution of damage, but the underlying mechanism remains the same: reduced efficiency due to structural loss. There is no reversal of this architecture. Neurons that have been lost do not regenerate in a way that restores original networks. White matter pathways that have been disrupted do not reform with the same precision. The brain adapts, but adaptation is not restoration. What remains is a system that can still function, but with limits that were not present before. It requires more effort to maintain stability. It is more sensitive to additional stress. It has less capacity to compensate for new disruptions. The process that led to this state did not begin with a visible event. It began with instability during sleep loss, progressed through repeated microvascular interruptions, and accumulated without detection. Each stage built on the previous one, moving from functional disruption to structural change. The absence of early symptoms allowed the process to continue. The brain did not signal when small regions were damaged. It adapted instead, maintaining outward function while internal structure shifted. By the time the effects become measurable, the underlying changes are already established. This is the defining pattern of silent strokes and sleep loss. Damage occurs before awareness. It accumulates without interruption. And once it reaches structural finality, it does not reverse. The brain continues forward in a new configuration, shaped by what has been lost rather than restored.
Pathology of Fear is a medical documentary series presenting clinically documented neurologic and pathological mechanisms for educational analysis.